Hello, everyone. My name is Alana Davis, and I am with Yasmin Gonzalez, Alexa Torres, and Cameron Biggs. We are from Professor Condoyan's Biology 104 Genetics Cell Lab of the fall 2022 semester. Our presentation is on obesity, specifically the SNP RS2229616 and the melanocortin-4 receptor. To introduce this project, the goal was to analyze and interpret ancestry data in order to determine a trait or disease risk that this donor may be susceptible to or may already be present. To do so, we must look at two essential concepts, SNPs and an RSID. SNPs, or single nucleotide polymorphisms, are single base pair substitutions that occur within and outside genes 1 through 3. A reference single nucleotide polymorphism, or RSID, is a unique identifier specific to an SNP. It is often used in genetics to help organize and sort data so one can easily research through a database using the RS number ID. Next, we will look at obesity as the disease and risks. Obesity is a complex disorder affected by genetic and environmental factors. It is characterized by a body mass index, or a BMI, being greater than 30 kilograms meters squared. It often occurs when individuals have a higher caloric intake than their energy expenditure. Worldwide obesity has doubled from 1980 to 2008. In 2014, it was recorded that obesity had a prevalence of 13% in the adult population of the United States. With the presence of a family history of obesity, the risk of obesity in childhood increases and is most associated with the melanocortin-4 receptor, or MC4R gene.
Some background on our SNP. The function of the MC4R gene directly relates to the conservation of energy in the human body. When there is a strong presence of the gene, our body is better able to regulate energy for homeostasis and keep it from burning calories too quickly. Our SNP is a variant in the MC4R gene that has an indirect correlation with obesity and is associated with appetite regulation, BMI, and other features of the metabolic syndrome. Our methods. After selecting our SNP, we searched the two given datasets provided by Professor Condoyan and Dr. Ross to find the corresponding RS number. The ancestry datasets used were obtained voluntarily by saliva of anonymous individuals. They were tubed and shipped to the ancestry company. Our RS number was found on the human ancestry dataset number 2. In the third step, we analyzed our sample by looking at what chromosome, position, and genotype it displayed. We then used SNPedia and OMIM to identify the genotype that is associated with normal expression of the MC4R gene. Next, we were able to look at a specific study to see if certain populations were more vulnerable to the mutated MC4R gene. After, we were able to give our anonymous donor advice based on their provided sample.
The SNP is linked to the 18th chromosome, located on the 58,039,276 position. The nucleotide sequence provided by the donor resulted in a CC genotype, as seen within the dataset. However, this genotype is not found within the summary of the phenotypic expression described in Figure 1.1. This is because the base reading within SNPedia read the complementary strand. So the CC genotype would correspond with the GG genotype since they are complementary of one another. However, there are two GG summaries found within the figure. The one we care about more is the bottom summary. The top GG summary simply states that it is the normal, most common genotype found within humans. The bottom summary indicates that there is a lower BMI and decreased obesity risk associated with the GG genotype compared to other genotypes on the SNP. The figure indicates the percentage of genotypes on the SNP from each population. As mentioned before, the homozygous G is the most common genotype and leads to lower BMI and decreased obesity risk. Only a small amount of individuals seem to have a homozygous A genotype. The heterozygous genotype of AG seems to be the second most common genotype, with similar expression to the homozygous A genotype. There's a slightly higher risk of metabolic syndrome occurring to those with the AG genotype, but not enough to consider the evidence conclusive. Since our donor had the most common genotype of CC, the origin of this person cannot be determined accurately through this gene alone, since there isn't much variability across the different human population.
We were able to predict that this individual is unlikely to have obesity after analyzing the nucleotides of their MC4R gene. They displayed a CC genotype, which is complementary to the GG nucleotide and is associated with normal or less of a risk of obesity. This is not to say that they should not maintain a well-balanced, rational diet and exercise. It is important for us to keep healthy habits and eat clean foods regardless of risk. However, individuals might face struggles finding a healthy relationship with food, seeking appropriate food portions, or stress without food. This can be due to the mutation in the RS2229616 that is highly associated with food regulation. Lastly, we can predict based on the ancestry datasets that this donor was a male. Males are often considered to have a lower risk of obesity than females. This experiment has allowed us to apply our learnings of SNPs to real-life situations. It is relevant to each of our career goals in the medical field. Most importantly, it allowed us to better understand the likelihood of diseases that run in our family and how these SNPs can affect us. We would like to acknowledge Sose Kendoyan and Dr. Ross for overseeing and providing the materials to conduct our research. And this is our literature cited. Thank you.